Okay, so RUNX1 codes for an important protein, which determines how many genes that are essential for the blood development process are actually regulated. And there's a number of different mutations that arise in this gene, and these lead to different disease outcomes, such as familial platelet disorder and acute myeloid leukemia, or AML. And there's a few different studies that have been done to try and understand how these different mutations cause the different diseases. For example, the generation of mouse models. And it's still unclear exactly what these proteins are doing within the cells, and so why the clinical outcome is so different. We know, for example, that some mutations can be inherited, whilst others completely block blood development, so only arise later. And to try and work out this question in the study, we used embryonic stem cells, where we could induce the mutant proteins at a specific time. And then we cultured the embryonic stem cells in a way to turn them into blood and switch the mutant proteins on. And this allowed us to then study the first events exactly after the mutant protein is there in the blood progenitor cells. And we did this genome-wide to look at the molecular function of the proteins. And quite surprisingly, even at this very earliest stage, we found that the response was very different between the different mutant proteins. And it was these differences that we then explored. So the main question that we get, and we've had all along, is really how do mutations in just this one single gene cause such big differences in the patients? And there's a couple of aspects to this. One is that RUNX1 is involved in multiple cell types.